Hello students. So today we are going to learn third part of excretion and osmoregulation. In the first part we learned the modes of excretion, that is ammonotelism, ureotelism, uricotelism, guanotelism. In second part we learned the excretory organ, that is kidney, ureter, urinary bladder, urethra, right? Now in today's part we are going to learn about urine formation, composition of urine, role of other organs in excretion, disorders and disease, hemodialysis, peritoneal dialysis, kidney transplantation. Now, from this last 2-3 parts which you are going to learn will be useful everyone. The reason, the disease disorder which is presently seen, the major one being the kidney stone, right? So where it is formed, how it is formed. Secondly, dialysis, which is also one of the major factor presently. There are quite a few number of percentage of population having uh, diabetic and highly diabetic and need of dialysis. So again, two types are there. And thirdly, the kidney transplantation. So here there's a very good video, or you can say short animation video, which shows how the transplantation is carried out, how it is the surgery is carried out, which part of the kidney, and how the earlier kidney remains in its original position, and which is the position of the new kidney transplantation. Okay, now in the present slide, as always, you can see something related with the topic. So you can see here nephron, right? The structural unit of kidney, and you can see very clearly the purification process taking place, where through afferent arteriole the fluid coming inside and moving out by the efferent, and how the other unwanted particle which is coming out through the PCT, that is the urine, further passing to Henle's loop, then towards the DCT and the collecting tubule. Finally it is expelled out through the ureter into the urinary bladder and through the urethra. Okay, and just few other things. If you see on these sides, solute keys, that is in the blood, what all things may be present: RBC, water, glucose, urea, sodium ion, potassium ion, chloride, bicarbonates, hydrogen, right? So other components present. And so what is the water intake? And again you can see the small chart coming out, which shows the amount of blood, whatever is present in the blood, or the because of it high salt diet, high blood pressure, all these things. And also towards the left you can see a meter reading and osmolality, glucose, urea, other content present in the urine. Moving on to the next slide, here the introduction part, urine formation. Process of urine formation is completed in 3 successive steps. That is, 3 major parts are there: ultrafiltration, which is otherwise also called as glomerular filtration, as it takes place in the glomerulus; selective reabsorption; third, tubular secretion or augmentation. So I put a simple image or chart showing how the blood or the purification takes place in the glomerulus. Through the afferent artery it enters, and unwanted stuff which is further passed into the PCT, and the purified one passes to the efferent arteriole, and so on. And also you can see the 3 types: glomerular filtration, tubular reabsorption, and tubular secretion. 3 tables given, or what is the process taking place. That is the movement of substance from blood within the glomerulus in the glomerular space, hence the name glomerular filtration. Tubular reabsorption, the movement of substance from the tubular fluid back into the blood. There is important components which are taken back into the blood. Then tubular secretion, movement of substance from the blood into the tubular fluid. We come into the first part or first step, that is ultrafiltration or glomerular filtration. So again towards the right side you can see a very good animation image showing the glomerular filtration, how the from the efferent the fluid comes inside, or the blood comes inside, and by purification the unwanted stuff is moved into the neck towards the PCT, and from the other end to the efferent arteriole purified blood passes further, right? So we go into the detailed matter. First point, diameter of efferent arteriole is greater than the efferent. As you can see, the efferent is always larger, which the reason is because of the high pressure in which it flows. Diameter of capillary cyst is smaller than both arterioles. Due to such difference in diameter, blood flows with a greater pressure through the glomerular. That's why it is slightly bigger, that is diameter in case of efferent arteriole than the efferent. This is called as glomerular hydrostatic pressure, in short GHP, and normally it is about 55 mm per Hg. This pressure is opposed by osmotic pressure of the blood, that is normally about 30 mm per Hg, and capsular pressure normally about 50 mm Hg. Hg, as all of you know, that is mercury, is always measured with relation to mercury. Hence, net effective filtration EFP is 10 mm per Hg or 10 mm Hg. Now, how it is, just towards the right side, you can see I have put a small chart, or we can say small image showing how this takes place. That is, EFP is equal to hydrostatic pressure in glomerulus minus osmotic pressure of the blood plus filtrate hydrostatic pressure, which is 10. That is, the hydrostatic pressure, as you can see, it is 55 minus 30 plus 15. So when you do the calculation, you get EFP as 10 mm per Hg. Further, walls of capillaries are extremely thin. Under the effect of high pressure, walls become permeable to major components of blood, except blood cells and macromolecules like proteins. That means, through as it is effect of high pressure, the walls are permeable to all the components, so that is, it can reabsorb or it can absorb. So only exception, blood cells and larger molecules like protein. That's why the macromolecules, they are not reabsorbed over there. Further, the plasma, except proteins, oozes out through the walls of the capillary. About 600 ml blood passes through each kidney per minute. So you can imagine, in per minute or 1 minute, 600 ml of blood passes through the kidney. The blood plasma flowing through kidney is filtered as glomerular filtrate, as it takes place in the glomerulus at the rate of 125 ml per minute, or if you calculate in a day, it is 180 liters per day. As you can see, 125 ml per minute, that is 60 seconds, right? So if you calculate in one day, it is 180 liters. Glomerular filtrate, deproteinized plasma. Primary urine is alkaline, contains urea, amino acids, glucose, pigments and inorganic ions, right? So here you can see what are the main contents: urea, amino acids, glucose, pigments and inorganic ions, which is passed on through the glomerular filtrate. Glomerular filtrate passes through filtration slits in the capillary space and then reaches the proximal convoluted tubule, that is the PCT. Moving further, selective reabsorption. Second part, the PCT is a place of reabsorption. It is highly coiled, so the glomerular filtrate passes through it very slowly. Columnar cells of PCT are provided with microvilli, due to which absorptive area increases enormously. This makes the process of reabsorption very effective. That is, as the name is selective reabsorption, it selectively reabsorbs important component which may escape out. Now, because of the microvilli, the absorption area increases and it can easily reabsorb the contents which escapes out during the filtration. These cells perform active, that is ATP mediated, and passive simple diffusion, that is reabsorption. Substances which considerable importance, that is high threshold, like glucose, amino acids, vitamin C, calcium, potassium ions, sodium ions, chlorine ions, are absorbed actively against the concentration gradient. Low threshold substances like water, sulfate, nitrate, etc. are absorbed passively, that is through the simple diffusion. In this way, about 99% of glomerular filtrate is reabsorbed in PCT and DCT. That clearly means that most of the things are reabsorbed, all the important components in the glomerular filtrate through PCT and DCT. Here I have put up a simple chart showing the reabsorption, where you can see the different labeling: thin limb, thick limb, that is of nephron loop, that is you can say the Henle's loop, descending lymph, then capsular space, glomerular space, which is there. Again, it is separately shown. Efferent, you can see the arteriole slightly larger with the larger diameter than the efferent arteriole. Then proximal convoluted tubule, you can see how the inner lining lies. Renal, that is the renal tubule. Then further the descending lymph, ascending lymph of the loop, then distal convoluted tubule, that is DCT, further moving into the collecting tubule. In the right side, again I kept a simple animation, or you can say GIF, where you can see how through the glomerulus, once the blood plasma enters in the efferent, moves to the efferent after the filtration, but remaining components, it passes through the neck into the PCT, and then the Henle's loop, descending and ascending, and further DCT and the further part, that is the collecting tubule. So you can see here very clearly the capillary tube which is present, which you can see with the different coloration: the red, which is sodium, the green, which is glucose, and yellow, which is amino acids, which helps to understand the concept clearly. Further, third part, there is tubular secretion, or otherwise called as augmentation. Finally, filtrate reaches the distal convoluted tubule via loop of Henle. Peritubular capillaries surround DCT. Cells of distal convoluted tubule and collecting tubule actively absorb the waste like creatinine and ions like potassium, hydrogen from peritubular capillaries and secrete into the lumen of DCT and CT. So these components like creatinine, ions, which is mainly potassium hydrogens, they are secreted further into the lumen of DCT, CT, that is distal convoluted tubule and the collecting tubule, thereby augmenting the concentration of urine and changing its pH from alkaline to acidic. That is why the tubular secretion is also called as augmentation. Secretion of hydrogen ions in DCT and CT is an important homeostatic mechanism for pH regulation of blood. This process is called as tubular secretion or augmentation. Tubular secretion is only mode of excretion in marine body fishes and desert amphibians. So this particular secretion is the main mode in marine bony fishes and desert amphibians. Further, we come to the concentration of urine. Again, if you see the chart, you can see 300 ml of osmolarity, or then further 600 ml osmolarity, 900 ml osmolarity, and 1200. And you can see the coloration also, how the coloration shows, which shows the concentration. There is from the efferent, different Bowman's capsule, glomeruli, where water, sodium is again reabsorbed, or in the low middle layer where it is partially, you can see it is red and blue, and lower part is completely bluish. Coming to the written matter, that is under the conditions like low water intake or high water loss due to sweating, human can produce concentrated urine. That's why the concentration increases. What can be the reason? Either due to excessive sweating, there is loss of water, and it can be due to low intake of water. That can be the other reason. It can be almost 4 times concentrated, that is 1200 ml, or you can say osmolarity. It is given as OSM, means osmolarity, than the blood, which is 300 osmolarity. For this purpose, a mechanism called as countercurrent mechanism is operated in human kidneys. Limbs of Henle's loop, that is juxtamedullary nephrons and vasa recta, operate countercurrent mechanism as follows. Now in the previous video we have learnt about the juxtamedullary nephron, where it is present, exactly which position, which helps to understand the present concept. Further, the mechanism involves the passage of fluid from descending to ascending lymph of the Henle's loop. Flow of tubular fluid is in opposite direction through both limbs, hence the name counter current or opposite flow. That is why this mechanism also called as counter current. Again, what is the reason? As you see, both the limbs, the descending and ascending, functions in the opposite direction, right? Or the flow in the opposite way. Hence, it is counter current mechanism. In case of vasa recta, blood flows from ascending to descending parts of itself. Wall of descending lymph is thin and permeable to water, means it takes up water readily, whereas that of ascending lymph is thin and impermeable to water. That which makes us understand very clearly. Further, in this region of descending lymph, water diffuses with tubular fluid into tissue fluid, due to which tubular fluid becomes concentrated. That's why if you see the later part, the concentration is high, that is in the DCT and the collecting tubule part. Ascending lymph of Henle's tube is thick walled and its cells can reabsorb sodium and chlorine ions from tubular fluid and release into tissue fluid. Due to this, tissue fluid around descending lymph becomes concentrated. This makes the water or more water to move out from descending lymph into the tissue fluid by osmosis. That's why you read it is permeable to water, that is the descending part. Thus, as tubular fluid passes down through descending lymph, its osmolarity, this concentration increases gradually due to water loss, and on the other hand progressively decreases due to sodium and chlorine secretion as it flows up through the ascending lymph. Besides, whenever water retention is necessary, pituitary secretes ADH. Now this ADH helps to retention of the water. ADH makes these cells in the wall of the collecting ducts permeable to water. Due to this, water moves from tubular fluid into tissue fluid, making the urine concentrated. Cells in the walls of deep medullary part of collecting ducts are permeable to urea. As concentration of urine flows through it, urea diffuses from urine into tissue fluid and from tissue fluid into the tubular fluid through thin ascending lymph of Henle's loop. This urea cannot pass through the tubular fluid while flowing through thick segment of ascending lymph. DCT and cortical portion of collecting duct due to impermeability for it in this region. However, while flowing through collecting duct, water reabsorption is operated under the influence of ADH. Due to this, urea concentration increases in tubular fluid and some urea again diffuses into tissue fluid in deep medullary region. Thus, same urea is transferred between segments of renal tubule and tissue fluid of inner medulla. This is called as urea recycling. Just as we read, urea concentration increases in the tubular fluid. So when it increases, some urea is again diffused in the tissue fluid, which is mainly in the medullary region. Operated for more and more water reabsorption from the tubular fluid and thereby excreting small volumes of concentrated urine. Now, from the concentration of urea or urine, we move to the further part, that is osmotic gradient, which is related with the earlier part only. Osmotic gradient is essential in the renal medulla for water reabsorption by countercurrent multiplier system. This osmotic gradient is maintained by vasa recta by operating countercurrent or exchange system. That's why, just as we learned the countercurrent mechanism, that is opposite flow in the ascending and descending. As we can read further, vasa recta also has descending and ascending limbs. Blood that enters the descending lymph of the vasa recta has normal osmolarity of 300 osmolarity, as here we can see osmolarity of 300 ml. As it flows down in the region of renal medulla where tissue fluid becomes increasingly concentrated, sodium, chlorine and urea molecules diffuse from the tissue fluid into the blood, and water diffuse from blood into the tissue fluid. You can see here happening vice versa. Due to this, blood becomes more concentrated while now flows through ascending part of the vasa recta. This part runs through such region of medulla where tissue fluid is less concentrated. Due to this, sodium, chlorine and urea molecule diffuse from the blood into tissue fluid and from water from tissue fluid to blood. This mechanism helps to maintain the osmotic gradient. Now earlier we read it was the other way around. Here it is again the water from the tissue fluid to the blood moves back. So that's why the osmotic gradient is maintained because of this counter current mechanism. So as to reabsorb water to maximum capacity, loop of Henle are longer in desert mammals like camel. Due to this, camel excretes concentrated urine. And as the best example here in case of camel, as a desert species or other desert animals, as they get less water, they have to preserve water because of the desert condition. Availability of water is less, so they excrete concentrated urine. That's why I put an image related to it. Can see a camel can survive for several days without drinking water even in high temperatures. How it is possible? Because they have got several specific features that enable them to survive the extreme conditions they encounter in the desert. These conditions are like fluctuating core temperature can be as low as 34 and as high as 41. As we know, in the daytime the temperature is very high, whereas in the later in the evening it starts to decrease. Large roundish body, fat concentration in the hump and extremely thin legs, which is the second factor. A slower metabolic and breathing rate in summer. When breathing rate is slow, the water evaporation through breathing will also be lesser. Blood with a higher water content. So blood contains more water. Extremely dry feces. Even the feces which is excreted does not have water content or very less. Lying down for long periods during daytime and urinating down its legs. So through the legs it's urinate. That's other reason how it keeps the body cool. The water excreted is concentrated. So these are the different ways of surviving in the desert condition. Towards the right side, you just have put a simple figure of the medullary osmotic gradient, where you can see from the tubular fluid, the proximal tubule, the direction of flow into the Henle's loop, where water which is reabsorbed, and further from the descending to the ascending loop, the potassium and sodium ions are reabsorbed, and further into the distal, from the distal into the collecting tubule, and finally into the renal pelvis. Further, composition of urine. Now again it is a important aspect. Normal urine is pale yellow colored transparent fluid due to pigment urochrome. So as we know, the urine coloration is pale yellow. That is because of the pigment urochrome. Now just to add over here, in case if it is dark yellowish color, what does that mean? The secretion of urochrome is higher, or if it is less, then the pigment secretion is lesser, which indicates certain type of disease or disorder in the body. That is why always our urine test is always 100% accurate. We can come to know if any disease or disorder is there. If you do the urine test, we can easily come to know very quickly rather than the blood test. Okay, moving further, composition of urine depends upon food and fluid consumed by the individual. There are two ways, one by regulating water reabsorption through ADH, and other by electrolyte reabsorption through RAS. Now firstly, so the urine coloration or what is present in the urine depends on types of food we are consuming and fluid intake also, right? And here there is another term given as RAS. What is this RAS? It is renin angiotensin aldosterone system, which is given as RAS. Okay. Further, hypothalamus in midbrain has special receptors called osmoreceptors. They can detect change in osmolarity, that is measure of total number of dissolved particles per liter of solution of blood. If osmolarity of blood increases due to any reason, such as after eating namkeen. Now I think most of them have the habit of eating namkeen, salty food, or whichever food we are seeing, to preserve the content. All the packaged food has got a lot of salt content, right? And usually in the evening snacks we usually munch in the namkeens more often, or due to sweating. In other words, due to water loss from the body, osmoreceptors trigger release of antidiuretic hormone, that is ADH, as we are learning earlier, from neurohypophysis posterior body. Here I will just add to it, other than namkeen. Now those who consume lot of junk food, again in junk foods salt content is always higher. Okay, moving further, ADH stimulates reabsorption of water from large part of DCT and entire collecting duct by increasing the permeability of the cells. This leads to reduction in urine volume and decrease in osmolarity of the blood. Once the osmolarity of the blood comes to normal, activity of the osmoreceptors cell decreases, leading to decrease in ADH secretion. This is called as negative feedback. That is, if the activity of the osmoregulator cells decreases, then the secretion of antidiuretic hormone also decreases, which is, we can say again here, the reverse process happening, or negative feedback, which is called as. In case of hemorrhage or severe dehydration too, osmoreceptors stimulate ADH secretion. ADH is important in regulating water balance through kidney. So ADH performs an important role in maintaining the water balance. In the absence of ADH, diuresis, there is dilution of urine takes place and the person tends to excrete large amount of dilute urine. This condition is called as diabetes insipidus. That is why in diabetic condition, which is we can see more often, the person feels to urine it quite often, even though if he may be drinking less amount of water, still every now and then he feels like urination. Why? Because of absence of ADH. What is that condition called as? Diuresis. Okay. Then another regulatory mechanism, RAS, just as we learned in the beginning part, repeat the full form, renin angiotensin aldosterone system by juxtaglomerular apparatus, JGA, which we learned in the previous part of the video. Whenever blood supply to efferent arteriole decreases, example low BP, dehydration, JGA cell release renin. Renin converts angiotensin secreted by hepatocytes into liver to angiotensin. Angiotensin converting enzyme modifies angiotensin 1 to angiotensin 2, which is the active form of the hormone. So just if you see in simple words, as I just read, low BP, dehydration, what happens? GGA releases renin. Renin converts angiotensinogen to angiotensin. Angiotensin is further converted to angiotensin 1 to further angiotensin 2, which is the main active form of the hormone. Further, now as we are learning about angiotensin 2, which is active form, just going into little detailed part of it, has triple function. Angiotensin 2 has triple function, that is, it constricts arterioles in kidney, thereby reducing blood flow and increasing blood pressure. Stimulates PCT cells to enhance reabsorption of sodium, chlorine and water. It stimulates adrenal cortex to release another hormone called as aldosterone that stimulate DCT and collecting ducts to reabsorb more sodium and water, thereby increasing blood volume and pressure. So you can see three important functions. That is why it is called a triple function. One, reducing the blood flow, increasing blood pressure, then reabsorption of sodium, water and chlorine, then another hormone which is released, that is aldosterone. Clear? Further, both ADH and RAS are essential. So both are important. Only ADH can lower blood, that is sodium reabsorption, thus maintaining osmolarity of the body. Whereas action of ADH and RAS leads to increase in blood volume and osmolarity. A large increase in blood volume and pressure stimulates atrial wall to produce atrial natriuretic peptide. See another important term, ANP. What is ANP? Atrial natriuretic peptide. Now what is that? See further. ANP, that is atrial natriuretic peptide, inhibits. What is inhibits? We can say stops sodium and chlorine reabsorption from collecting ducts, inhibits release of renin, reduces aldosterone and ADH release too. This leads to a condition called as natriuresis. Increased excretion of sodium in the urine. That is, more amount of sodium is excreted through the urine. When? Because of A and B, and diuresis caused. If it is diuresis, then what it is? Diabetes insipidus, where frequently urine secretion, the person feels to urinate. Further, kidney participate in synthesis of calcitrol, the active form of vitamin D, which is needed for absorption of dietary calcium. So you can see here the calcium which is present in the body, this absorption is required because of vitamin D. That's why vitamin D absorption is also required. Otherwise, if vitamin D absorption is less, then again absorption of the dietary calcium into our body will be less, which again can carry out various other problems related to bones. See further part, deficiency of this calcitrol or synthesis, as we have just read, can lead to brittle bones or weak bones. That's why every now and then the person may have a fracture in the bone, or when falls down, pain in the bone or dislocation of the bone, etc. In the right side, I put a simple chart showing what we read in the previous slide, how the stage wise process takes place. As we read, angiotensinogen, which is converted to angiotensin 1 by the renin, angiotensin 1 into angiotensin 2, which is the active form, which we have read out just now. So in that, AT1 receptor, which releases to kidney, heart, vasculature, central nervous system. Further, kidney to aldosterone, sodium and water retention takes place, where in case of heart, proto-oncogenes, growth factors, cardiac hypertrophy, remodeling, or if I just use the term oncogenes, which is usually referred to the cancerous genes, right? Then vasculature, that is vascular constriction, that is our blood vesicle they constrict. Increased peripheral resistance. Further, the CNS2 reactive oxygen species ROS or sympathetic nerve activity. Further, neuronal inflammatory response. So here hypertension, heart failure, atherosclerosis or neurodegeneration, stroke, all these can happen. In case of peripheral, I repeat, if it is peripheral part which is affected, hypertension, heart failure, atherosclerosis, which more often is happening, that is heart failure, and all are, we can say, interrelated. And in case of central nervous system, it is a neurodegeneration, degeneration of the brain, functioning of the nervous system, etc., or stroke. Now moving further, little bit of additional information towards the left side, we can see the interesting fact about the bat. So first we'll just go through the information. Vampire bat. This you might have seen in the movies, cartoons, etc. Talking a lot about the vampire bat, right? From South America is a nocturnal sanguivorous mammal. What is sanguivorous? I hope you know, we are learned in the chapter classification, means blood-sucking mammal. Right, for example, if I give another example, leech, right, it is blood sucking annelid, or very simple bed bug, mosquito, blood sucking arthropod insect, right. Okay, it has to fly long distance to locate suitable prey. Once found, it can even consume blood to an extent more than half its body mass. In such a case, the body of the bat becomes too heavy to fly. So the bat has to fly a longer distance to find a prey for blood sucking. Once it finds, it sucks the blood as much as possible, more than its weight, half the weight. To compensate for this, while the bat is feeding, its kidney excrete large amount of dilute urine, up to 24% of its body mass. Now bat can fly back to cave, tree, wherever it spends its time, where it can spend the day. During the daytime, it cannot go drink water. At the same time, diet being high on protein, large amount of nitrogenous waste is produced. So instead of dilute waste, kidney resort to concentrated urine in order to conserve water. This capacity to rapid change of osmolarity of urine is a classic example of adaptation. So in the right side, I had put a very good animation image of the vampire bat, right? And above again South American vampire bat, which is very common, but it is as a blood sucking bat which preys on larger mammals. While taking a blood meal, its kidney excrete large quantity of dilute urine. It is now lighter for flight, means it helps to fly easily if the dilute urine is secreted. Otherwise, it will be very difficult to fly. While roosting, the bat conserves water by excretion, small amount of highly concentrated urine. Now, moving further, now from kidney, the urine formation, now we come to other organs in our body which helps in excretion, like skin. So first part, skin of many organs in this is thin and permeable. It helps in diffusion of waste product like ammonia. Human skin is thick and impermeable. It shows presence of two types of glands, namely sweat glands and sebaceous glands, which we are learning from the earlier class as well, right? Sweat glands are distributed all over the skin. They are abundant in our palm region and facial region. That's why we sweat more in the facial and the palm region. These simple unbranched coiled tubular glands open on the surface of the skin through an opening called as sweat pore. Sweat is primarily produced for thermoregulation. What does thermoregulation means? To maintain the body heat. But it is primarily produced for thermoregulation, but it also excretes substances like water, NaCl, lactic acid and urea along with the sweat. Okay, then sebaceous glands second, they are present at the neck of the hair follicles. They secrete oily substance like sebum. It forms a lubricating layer on the skin, making it softer. It protects skin from infection and injury. So as you know, even in certain persons, they may sweat a lot, even the skin looks oily, but actually this protects the skin from infection and injury. Towards the right side, I put the image of the skin diagram, human skin diagram. On the left side, epidermis, the uppermost layer, dermis, the layer beneath the epidermis, and subcutaneous tissue. Towards the right side, you can go in detailed part of the structure. Stratum, corneum, granular cell layer, spinous cell layer, basal cell layer. Further, the sebaceous gland, as we read out, we can see glands very clearly at the neck region of the hair. Sweat gland, then nerves, hair follicle, collagen and elastic fiber, artery, vein, and the fat molecule in the subcutaneous tissue. Okay. Further, lungs. Lungs are the respiratory organ, as again we learn in detail in the chapter respiratory system. They help in excretion of volatile substance like carbon dioxide and water vapor produced during cellular respiration. They also excrete volatile substance present in spices and other foodstuff. So if you are eating lot of spicy food, which most of them like, right? Spices in the food. So these are also excreted through the lungs. Further, coming to another important aspect, disease and disorder. So here I have shown some images which may be really what do you say? See, by seeing the image we can feel, how come this things can happen? That's why not always take care of a kidney, heart, lungs, all are important functional organ. If these gets affected, then it will be very difficult aspect for the person. That's why you have to always have proper food, hygienic food, exercise. All these things are necessary. So we go one by one with the disease and disorder. Firstly, introduction part of it, excessive albumin in the urine. What is albumin? In simple words, the protein content causes albuminuria or proteinuria. Indicates injury to the endothelial capsular membrane as a result of increased blood pressure. injury or irritation of the kidney cells by substances such as toxins or heavy metals present in the body, or even in case of bodybuilders, they take a lot of protein content, but if they are having proper exercise according to it, it will be good. If they are not doing exercise and taking a lot of protein, then it will put pressure on the kidney. Second point, pressure of excessive quantity of ketone bodies in urine may cause due to diabetes mellitus. Now we just learned the continuous urine secretion, dilute urine, which way we do depending on the case called as diuresis or diabetes insipidus. Here excessive quantity of ketone, if it is present, it causes diabetes mellitus. So as we know, there are two types of diabetes. One is diabetes insipidus and second is diabetes mellitus, right? Now, further, starvation or too little carbohydrates in the diet. Third point, presence of leukocytes. What is leukocytes? The white blood cells, right? In urine indicates possibility of infection of kidney or other urinary organs. So if leukocytes are present in excess amount, it can cause infection in the kidney or the urinary organ. So if you in the right side, urine examination for anorexia nervosa affecting kidney. So it can be proteinuria, blood in the urine, 24 hours creatinine clearance test has to be conducted for that. Now if this creatinine level is high in the body, it can be very dangerous. As I told, it can be one of the reason for diabetes. Also right side you can see I put an image showing polycystic kidney. We come back to the polycystic. As you know, poly means many, cysts small nodules, right, which usually sometimes forms outside our body, that is in the skin, body region, etc. But you can imagine it can happen in our kidney also, which can affect the functioning of kidney very much. Now coming to the detailed part of the types. First is kidney stones. Now you can see on the right side of the image the stones which are present, how it once it forms, it will be difficult while secreting the urine or pain during the urination, right? Then you can see the lower part. I put a very simple image. Why? You can see the kidney stone, how it is originating from the renal calculi, from the kidney main part, right? Then based on that, you can see where all the kidney stones can affect depending on the size of the kidney. So it can be either in the renal calculi, or it can be in the renal pelvis, or it can be in the ureter or in the urinary bladder. Now usually this kidney stone is otherwise also called as renal calculi, as you can read in the first point of it. They may be formed in any portion of the urinary tract from the kidney tubules to external opening. Depending on that, they are classified into different types of kidney stones. Before that, just to add few more terms which are not mentioned over here, but for your knowledge, that is nephrolithiasis, which means what? Condition of having kidney. So if the kidney stones are present, the condition is called as nephrolithiasis. Now if the kidney stone is present in the urinary tract, it is called as urolithiasis. Okay, and if it is present in the ureter, it is called as ureterolithiasis. So it based on the position also different names accordingly. I repeat the terms: nephrolithiasis, that is presence of kidney stones; urolithiasis, presence of kidney stones in the urinary tract; and ureterolithiasis, that is presence of kidney stone in the ureter. Okay, as we saw just the motion of the kidney stone to the various part of the region in the kidney. Now coming to the type of kidney stone, right, based on what they compose of. First, it can be calcium stones. That is usually are calcium oxalate stones or calcium phosphate. One. Second, struvite stones. These are formed in response to bacterial infection caused by urea splitting bacteria. These grow very quickly and they become in quite larger also. That's where the problem arises when it blocks the urine or while urination, pain in the during the urination. Then Third is uric acid stones. In people who don't drink enough water or consume high protein diet can suffer from this type also. So that's why drinking at least 3 to 4 liters of water in a day is essential, because it will either dissolve or remove, does not allow the formation. And also those who take high protein diet should also be very careful, because of the high protein diet the kidney stone formation can take place. Then fourth, cystine stones. It is a genetic disorder that cause kidney to excrete too much of certain amino acids. So it is actually genetic, more often. Then symptoms: intermittent pain below the rib cage, in the back and sideways, hazy brownish reddish pinkish urine, frequent urge to pass urine, pain during micturition, as micturition means urination. See, these are the some of the symptoms. So best you can see the coloration of the hazy brownish reddish pinkish, which again indicates certain symptoms. As I told in the beginning, the coloration of the urine is because of urochrome, pale yellow color. If it is light or dark, it indicates disease or disorder. Then second, diagnosis: uric acid content of blood, color of the urine, kidney x-ray, sonography of the kidney are different diagnostic method depending on what is the symptom. Now coming to the second tab, uremia. Blood normally contains 0.01 to 0.03% urea, but if the rise above 0.05%, it is called as uremia. There is excess amount of urea in the urine. It is harmful because it may lead to kidney failure also. Third, nephritis. It is inflammation of the kidney characterized by proteinuria, just as we had albuminuria, proteinuria, caused due to increased permeability of glomerular capsular membrane, permitting large amount of proteins to escape from the blood to urine. That is, whenever there is high protein content in the food, this leads to change in blood colloidal osmotic pressure, leading to movement of fluid from the blood to interstitial space. It is reflected as edema. Or I'll just add few more terms over here. Other name of nephritis, it is glomerulonephritis, as it is moreover found in the glomerular region, or it is also called as Bright's disease. Okay, now coming to the fourth one, renal failure. It is a decrease of cessation of glomerular filtration. It is classified into two types: acute renal failure, in short ARF. ARF is sudden worsening of renal function that most commonly happens after severe bleeding. This is decrease in the urine output, oliguria, that is scanty urine, less than 400 ml per day, or less urine 0.5 ml per kg per hour in children. So you can see if the level is less than 400 ml in case of mature individual, and in case of children's 0.5. Other causes of ARF may include acute on obstruction of both ureter and nephrotoxic drugs. ARF can be detected biochemically by elevation of creatinine level. So again, if creatinine is level, it is one of this reason. Then chronic kidney disease, CKD. Second type of renal failure. It is progressive and generally irreversible decline in the glomerular filtration rate, that is GFR. It may be caused due to chronic glomerulonephritis. It can be detected by reduced kidney size and possibility of anemia. Now moving on to the next, there is hemodialysis. Now, which is now more commonly, you can see more patients are requiring dialysis because of the kidney failure. Okay, quickly moving into the points. When renal function falls below 5 to 7% accumulation of harmful substance in the blood, and substance in the blood begins. In such a condition, the person has to go for artificial means of filtration of blood. In hemodialysis, blood is filtered outside the body using dialysis unit. As we can see, the kidney failure leads to the dialysis, or you have to wait for kidney transplantation, right, which as we had learned in the previous video, where 2 lakh patients every year require kidney transplantation, but only 5,000 to 6,000 kidney transplantation takes place because of the shortage, because it is not possible, because of lot of reasons are there. Later on, in the later part of the video, we can see about how the kidney transplantation takes place when it is available with the donor. Okay, further, in this procedure, patient's blood is removed generally from a radial artery. It is passed through a cell pain tube that acts as a semi-permeable membrane. The tube is immersed in a fluid called dialysate. As you can see in the image, dialysate is isoosmotic to normal blood plasma. Hence, only excess salt, if present in plasma, pass through the cell pain tube into the dialysate. Waste substances being absent in dialysate move from blood into the dialyzing fluid. Filtered blood is returned to the vein. In this process, it is essential that anticoagulant like heparin is added to the blood while it is passing through the tube and before resending into the circulation. Adequate amount of antiheparin is mixed. Why? As we know, if blood clots in between anywhere, then again there can be heart failure problem towards the heart. That's why they have to add the anticoagulant, which does not allow for clotting factor. In the present scenario, you might have heard lot of heart attacks because of the coagulation. So they have to add anticoagulant, that is heparin, which prevents coagulation. So it has to be taken care while the circulation of the blood and also before the blood is again passed into the human body. The blood has to move slowly through the tube and hence the process is slow. That's why the dialysis process may take 2 to 3 hours. Okay, just see the figure. Now, as I told, it begins from the radial artery where the blood flows from it, and you can see the arrow process where remove blood for cleaning. Further, the arterial monitor pressure is there, pressure monitor is there. Then the blood pumping takes place. Heparin is added. Dialyzer inflow with the further pressure is monitored because it has to go very slowly. Then using dialysate, then dialyzer, then fresh dialysate, then further the venous pressure monitor to check the flow of blood, air trap and air detector if any air bubbles are formed, which again can cause heart attack. Then clean blood, which is passed into the body again. Now, again here there are different types. So here you can see peritoneal dialysis. What is it? First again we can go through the points. In this method, the dialysing fluid is introduced into the abdominal cavity of or peritoneal cavity. As you can see the position, the peritoneal membrane acts as semi-permeable dialysing membrane. Toxic waste and extra solutes pass into the fluid. This fluid is drained out after prescribed period of time. Peritoneal dialysis can be repeated as per the need of the patient. It can be carried out home, at work or while traveling. So it is a easy process. The patient can easily utilize it while traveling, while at work or at home. So it is an easy process, unlike the hemodialysis, which is not as efficient. You have to get admitted in the hospital and then carry out the dialysis. Kidney are associated with secretion of erythropoietin, renin and calcitriol, which is not possible using dialysis machine. I put both the images, animation image, to understand about how hemodialysis takes place, how peritoneal. See the left image, the hemodialysis. The first step, which is very common, which is very important for the critical patient or where the hemodialysis has to be taken. As just now we saw how the process takes place from the radial artery, pressure monitor, then moving further, blood pump, heparin pump for stopping from coagulation, waste removal, then further cleaning, dialysate, then pressure monitor, air trap monitor, and then it is pumped back into the blood. So it is a process which is required 2 to 3 hours. Whereas peritoneal dialysis is very simple, which can be used while traveling, while at work, while at home, you can easily carry out. But in severe cases where the dialysis has to be taken with complete care, hemodialysis preferred. In a lighter case, the peritoneal dialysis can be easily carried out. Now, kidney transplantation. See the right side, how it is carried out. Very simple way how the transplantation is carried out. So you can see how the connection is made, how the joint is carried out, how the surgery is done for kidney transplantation. So very good animation video which you can understand how it is done. So specifically for a medical student, for MBBS students, so it is very good to understand how it is carried out, and even for a normal person or all students, it is easier way to understand how this is carried out and how it is completed. It's not finished yet, till it completed, how it is cut open and how it is closed, right? And towards the left you can see living donor. So most probably if the child is affected, the mother, father, brother, sister are the donor, because it can match. Also see carefully in that image, in the body, the earlier kidney which has been infected is not removed. Why? Because it has been seen in the beginning of the transplantation, when the earlier kidney is removed, the kidney transplantation was not successful. So then later on they kept the original kidney in its position, and the new kidney which is transplanted is slightly fitted at the lower end, transplanted the lower end. You can see position also very clearly. Okay, otherwise the body system gets affected. So we can see the points. It is organ transplantation of a healthy kidney into a patient with end stage renal disease. Kidney transplantation is classified as cadaveric or a decayed donor if the donor is not living. So any patients, they died due to any reason, so immediately it can be transferred if the patient's health condition was good earlier. Or living donor, that is as I just mentioned, from the parents, for father, mother or brother, sister, or from the relative which is matched. Then living donor kidney transplant are further classified as genetically related, as from the family, or non-related, they are not family members, but their kidney transplant can be carried out if the conditions are fulfilled. And last, today's assignment question based on what we have learned. So thank you all of you. This is the end of the chapter.